When night falls, the skies above us become an invisible acoustic arena. For millions of years, while most creatures have relied on the fading light to navigate the world, one remarkable group of mammals took to the air, equipped with an extraordinary biological superpower. That superpower is echolocation. Bats, which account for more than 20% of all living mammal species, have conquered the darkness not by improving their eyesight, but by mastering the very physics of sound. Welcome to G Fun Facts Online, where today we are diving deep into the spectacular evolutionary puzzle of the bat skull. By emitting high-frequency vocalizations and meticulously analyzing the returning echoes, bats can paint a flawless three-dimensional acoustic image of their surroundings. This staggering ability allows them to weave through dense, tangled forests, dodge hair-thin obstacles, and snatch erratic insects from the air in absolute pitch blackness. But echolocation is far more than just a behavioral quirk or a simple parlor trick of nature. To project piercing ultrasound and receive faint, delayed echoes requires profound physical transformations. Over the span of tens of millions of years, the mechanical and acoustic demands of echolocation have fundamentally sculptured the anatomy of bats, completely altering their skeletal architecture. The evolutionary pressure to produce, direct, and interpret high-frequency sound waves transformed the bat cranium into a highly specialized bioacoustic instrument. By examining the fossil record, developmental biology, and high-resolution micro-CT scans of bat skulls, scientists are piecing together a story of how the interplay of sound, sensory perception, and eventually diet drove an incredible diversification of skull shapes, leaving us with a living testament to the power of evolution. To truly understand how echolocation shaped the anatomy of bats, we must travel back more than 50 million years into the Eocene epoch. The early stages of bat evolution have long been shrouded in mystery, primarily due to an incredibly sparse fossil record. Delicate bat bones do not fossilize easily. However, key discoveries have sparked intense debates among paleontologists regarding a classic evolutionary chicken and egg scenario. Which came first, powered flight or echolocation? One crucial piece of this ancient puzzle is Onychonicterus finii, a 52-million-year-old fossil bat discovered in Wyoming. Onychonicterus possessed primitive wings that indicate a style of flight much less advanced than that of modern bats. Initial anatomical analyses of its skull suggested that it lacked the enlarged inner ear structures typically associated with ultrasonic hearing. This observation led to the flight-first hypothesis, which is the idea that early bats evolved the ability to fly long before they developed the capacity to echolocate. However, in the world of paleontology, the debate remains fiercely alive. Recent re-examinations utilizing advanced computer-aided tomography have revealed tantalizing hidden clues. Scientists found specific bone connections in the throat and ear regions that perfectly mirror those seen in modern echolocating bats, strongly suggesting that Onychonicterus finii might have actually possessed rudimentary echolocating capabilities. The plot thickened with the discovery of Village Sige, a remarkably preserved 50-million-year-old uncrushed bat skull found in southern France. This fossil provided what researchers call the Rosetta Stone of bat echolocation. Because the skull was not flattened by millions of years of geological pressure, analysis of Villasia confirmed that its inner ear structures were highly akin to those of modern echolocating bats. This means that advanced, high-frequency echolocation had firmly evolved long before the origin of modern bat families. Interestingly, while we see widely varying abilities in adult bats today, with old-world fruit bats largely lacking laryngeal echolocation, the field of developmental biology offers a striking and undeniable revelation. Studies on bat embryos have shown that all bats, even the non-echolocating fruit bats, begin life in the womb with an exceptionally large cochlea. This embryonic structure acts as a living fossil hidden within their development, strongly suggesting that the common ancestor of all living bats possessed the ability to echolocate. In the lineages that eventually shifted strictly to visual foraging and eating fruit, this rapid cochlear growth simply halts during later fetal development. This fascinating developmental quirk demonstrates exactly how deeply the legacy of sound is inscribed into the ancestral bat blueprint. Between 58 and 34 million years ago, echolocation was the undisputed primary driver of skull shape evolution across early bat lineages. Think about the engineering required here. Emitting a sound that can travel through open space, bounce off a tiny, erratically moving target like a moth, and return with enough acoustic fidelity to be processed by the brain requires highly specific structural engineering. To achieve this, the bat cranium underwent significant alterations, specifically tailored to the exact frequencies the animals utilize. A sweeping analysis of hundreds of bat skulls housed in European natural history collections revealed a striking correlation. The overall size and shape of a bat skull have evolved to physically match the frequencies of its sonar. Bats that emit high-frequency sonar signals typically possess noticeably shorter faces and smaller skulls, but disproportionately wider ear bones. This is a direct physical reflection of their need to capture and process incredibly short wavelength echoes. Conversely, species utilizing lower-frequency echolocation tend to have larger skulls to accommodate longer wavelengths. The most profound divergence in skull morphology, however, stems from the literal exit point of the sound. Within the massive world of laryngally echolocating bats, there is a clear anatomical dichotomy. We have the mouth emitters and the nasal emitters. The ancestral and much more common approach to echolocation is oral emission, where bats project ultrasonic pulses directly through their open mouths. To optimize this acoustic pathway, mouth-emitting bats typically possess skulls with a distinct upward tilt. This craniofacial tilt allows the bat to fly with its head naturally angled to direct the sonar beam straight forward into its flight path, like a headlight on a car. Because the mouth serves the dual, competing purposes of vocalizing and catching prey, the snout region, known as the rostrum, must be engineered to rapidly snap shut on an insect the exact millisecond the echoes pinpoint its location. On the completely other side of the evolutionary divide are the nasal emitters. These include fascinating species such as the horseshoe bats and the leaf-nosed bats. These species hunt with their mouths completely closed, instead forcing their laryngeal calls up through specialized nasal passages and out through their nostrils. Transitioning from mouth to nose emission required a monumental redesign of the bat face. In nasal emitters, the palate is untilted to facilitate sound emission straight through the nasal cavity. More importantly, the internal nasal cavities evolved incredibly complex, maze-like bony structures called turbinals. These structures act as microscopic resonant chambers, fine-tuning and amplifying the sound before it ever leaves the bat body. Externally, this acoustic specialization is often adorned with elaborate noseleaves. These are fleshy, highly modified facial structures that act exactly like parabolic megaphones to focus and direct the ultrasonic beam into the night. For these specific bats, the skull shape and the complex internal chambers of the rostrum are heavily constrained by the strict physics of sound amplification and resonance. But the physical engineering does not stop at the exit point of the call. One of the most fascinating anatomical adaptations hidden beneath the skin of an echolocating bat involves the intricate connection between the apparatus that produces the sound and the apparatus that receives it. Most mammals, including humans, have a hyoid apparatus. This is a series of small bones in the throat that support the larynx and the tongue. In non-echolocating mammals and non-echolocating fruit bats, the stylohyal bone, which is part of this throat apparatus, hangs freely, completely separated from the ear bones. However, in laryngally echolocating bats, evolution forged a totally unique physical bridge. The proximal end of the stylohyal bone directly articulates with and is often completely fused to the tympanic bone, which forms the floor of the middle ear. This bony connection is absolute in laryngally echolocating species and entirely absent in all others, acting as a definitive skeletal signature for true echolocation. When a bat produces an ultrasonic call, the muscular contractions in its throat are incredibly forceful. This direct connection between the larynx support and the middle ear likely serves a crucial mechanical role. It may help coordinate the firing of middle ear muscles to dampen the bat hearing at the exact millisecond an incredibly loud call is emitted. This protects the delicate inner ear from self-deafening. Immediately after the call, the muscles relax to capture the incredibly faint returning echo. This exquisite anatomical synergy perfectly illustrates how heavily the invisible force of sound has influenced the physical bat skeleton. Of course, producing sound is only half the echolocation equation. The ability to hear and decipher high-frequency echoes requires profound neurological and morphological adaptations of the inner ear. The primary organ of hearing, the cochlea, underwent massive evolutionary expansion in bats. Compared to non-echolocating fruit bats, which average 1.75 turns in their spiral-shaped cochlea, laryngeal echolocating bats boast greatly enlarged cochlea that coil between 2.5 to 3.5 times. This extended spiraling vastly increases the length of the basilar membrane, allowing for a much higher density of microscopic hair cells that are perfectly tuned to specific, ultra-high frequencies. Recent comparative genomic studies have illuminated the complex genetic groundwork underlying these physical changes. Scientists have identified numerous genes related to hearing and auditory processes that underwent positive adaptive selection in both echolocating bats and toothed whales, such as dolphins and sperm whales. This is a classic textbook example of convergent evolution, where two completely different animals find the exact same biological solution to a problem. Genes governing cochlear bony development, ion balance, and auditory signal transduction mutated in parallel ways to support high-frequency ultrasonic hearing in these vastly different mammalian lineages. Specifically, sites in genes like TECPR2 evolved to enhance the functional capability of the cochlea, proving that the intense survival pressure to hear high-frequency echoes fundamentally altered bats all the way down to the molecular level. Beyond the gross morphology of the ear bones, the very wiring of the bat brain has been shaped by the specific type of echolocation utilized. The thousands of echolocating bat species broadly trace back to two distinct evolutionary lineages, often referred to as the yin and yang bats. A breakthrough anatomical study utilizing micro-CT scans revealed that these two lineages process sound using completely different neuroanatomies of the inner ear. In yin bats, the inner ear ganglion, which is the structure of neurons connecting the inner ear to the brain, follows a more constrained, traditional mammalian blueprint. However, the yang bats, which rely heavily on highly complex, frequency-modulating echolocation, evolved a completely novel anatomical configuration. They essentially removed the physical space constraint on the inner ear ganglion. This incredible structural freedom allowed the ganglion cells in yang bats to multiply extensively, creating a much denser network of neurons to process highly complex sound variations. This brilliant neurological remodeling is widely credited with driving the massive evolutionary diversification seen in the yang bat lineage today. While echolocation dictated the fundamental architecture of the bat skull for millions of years, an interesting and dramatic shift occurred approximately 26 million years ago. As bats expanded across the globe, they began moving into diverse ecological niches that offered an array of brand new food sources. While many bats remained strict insectivores, catching bugs on the wing, certain lineages, most notably the New World leaf-nosed bats, experienced an absolute explosion of dietary diversification. Over a relatively short evolutionary window, leaf-nosed bats evolved to eat fruit, drink nectar, hunt small vertebrates, and even consume blood. This presented a massive evolutionary conflict. How does a bat modify its skull to consume drastically different foods without ruining its precisely tuned acoustic equipment? If the skull changes shape to bite a hard fruit, does it break the sonar? Evolution solved this through a brilliant phenomenon researchers call decoupling. Sophisticated three-dimensional computational modeling of hundreds of bat species revealed that the evolutionary pressures of diet and echolocation began acting on different parts of the skull entirely independently. Because the upper portion of the skull, the cranium, is responsible for housing the brain, protecting the inner ear, and shaping the nasal cavities for sound resonance, it remained highly constrained by the rigid physical laws of echolocation. It could not change without breaking the biological sonar. The lower jaw, the mandible, however, is primarily tasked with feeding. The decoupling allowed the lower jaw immense evolutionary freedom. As a result, the lower jaws of bats morphed wildly to accommodate different diets. They elongated into thin snouts to lap nectar from deep flowers. They robustly thickened with powerful musculature to crack open hard, tropical fruits. or they modified into razor-sharp surgical instruments to delicately slice mammalian skin for blood feeding. In insectivorous bats that rely on capturing prey on the wing, the jaw and cranium co-evolved under a tight functional trade-off, balancing bite force with acoustic efficiency. But in the remarkably diverse leaf-nosed bats, the lower jaw could drastically change shape, while the upper cranium maintained its perfect acoustical integrity. The bat's skull is undoubtedly one of the most remarkable examples of evolutionary engineering found anywhere in nature. It stands as a profound testament to the fact that evolution is not merely about adapting to physical environments like climate or terrain, but also about mastering invisible forces. Through the relentless pressures of survival, the ethereal, invisible nature of sound became a literal sculptor, physically molding bone, tissue, and genetics over millions of generations. From the dramatic shortening of the face to accommodate high-frequency sonar, to the complex internal spiraling of the cochlea, and the intricate mechanical fusing of throat and ear bones, every single millimeter of a bat skull tells a story of flawless acoustic optimization. The evolutionary divergence into mouth and nasal emitters, the brilliant neurological rewiring of the inner ear ganglia, and the evolutionary decoupling of the jaw from the brain case to allow for extreme dietary variety all perfectly illustrate the profound plasticity of mammalian anatomy. Today, as bats perform their nightly silent symphonies across global ecosystems, pollinating our flowers, controlling dangerous insect populations, and dispersing seeds across degraded forests, they carry with them a 50-million-year-old architectural legacy. The evolution of the bat cranium reveals that, sometimes, the most profound and permanent changes in the physical world are driven by things we cannot even see. Sound did not just guide bats through the dark over the last 50 million years. Sound literally built them. Thank you for joining us today on G Fun Facts Online. Keep exploring, keep questioning, and we will see you next time to uncover more wonders of our incredible world.